Hello students, welcome to Trunkie Academy. We studied yesterday the chapter biotechnology, and in biotechnology we studied about the introductory part and the different the concept, the basic concept, and the important that what are the different important principles, okay, and what are different important process that involves the biotechnological process and biotechnological techniques, okay. Now today we'll study about the different tools, the tools of recombinant DNA technology. This is one of the important technology in biotechnology, okay, and this DNA technology has given a new dimension in the subject of biotechnology, okay. So we'll study about the tools of recombinant DNA technology. So what is this? Genetic engineering or recombinant DNA technology can be accomplished only if we have the key tools, that is the destruction enzymes, okay, that means to cut or to break into fragments of the DNA, and polymerase enzymes that polymerizes join, and then the polymerase, okay, DNA polymerase, okay. So that is the which enzyme which again forms the another strand of the DNA of single, when the DNA strand is the single strand DNA is there, okay. Ligases joins between two fragmented DNA segments, okay, ligases, and okay, so that is for the ligases, and the vectors which carry, and we can insert into a plasmid or in a DNA of other organisms, and then that can be entered into. So that way we is called the these are the important the things for the genetic or recombinant DNA technology. These are the important key tools or important things that is required in case of genetic engineering or recombinant DNA technology. Okay, so the host organism, vectors. We are using vectors, but at the same time we need the host organism on which we are doing this, for which we are doing we are applying this technology. Okay, so these are the important things or the key tools that we need in recombinant DNA technology, and how this acts and how this works. Okay, how these are required. These key tools work and how this is required. We'll see in the next few slides. Okay, so these are host organisms. Okay, so what do we do? We need destruction enzymes that cuts the DNA into fragments, polymerase enzymes that form the second strand of the DNA on single strand, ligases that joins the cut of the fragmented DNA, vectors which carry, in which we need to carry the alien DNA pieces, fragments, and then the host organism which we into which we insert and do the experiment. Okay. So these are the things that we require. Destruction enzymes means in 1963, the two enzymes responsible for restricting the growth of bacteriophage in Escherichia coli were isolated. Okay, the first time, and this discovery led to you can say a new started a new era in the science and the field of biotechnology. Okay, but this restriction enzyme plays a very important role in biotechnology. Okay, so in 1963, two enzymes responsible for restricting the growth of bacteriophage was observed and isolated. Okay, so this was the restriction enzyme. So one of these added methyl groups to the DNA methylase. Bacterial DNA is methylated, recognition site methylated, and restriction endonuclease does not part methylated site. So this was found that this restriction endonuclease or restriction enzyme. I told you yesterday while explaining about the biotechnology that restriction enzyme is actually the restriction endonuclease. So when bacterial DNA is methylated, this restriction endonucleases are not cutting the methylated DNA. So this was one of the point was observed. While other cut the DNA. In case of the other part, and if it is not methylated, it will cut the DNA. Okay, so restriction enzyme, restriction endonuclease is cut and cleaves into two cut, and the cut part is called sticky end because it is fragmented and it's ready to join with another part. Okay, so it's called the sticky ends after cutting the, okay, after becoming the fragments. The latter enzymes are called restriction endonuclease. Okay, the enzyme that cuts. I told you yesterday and explained yesterday. Okay? The first restriction endonuclease was reported by was the Hind2. The name was given as the Hind2. Okay, so whose functioning depends on a specific DNA molecule sequence. So it acts on a specific DNA site to cut it into the fragments. Okay, so that means the Hind2. Now Hind2 always cut DNA molecules at a particular point by recognizing a specific sequence of 6 base pairs. So restriction endonuclease are of different types, and the one which was the first was reported and discovered was the Hind2. Restriction endonuclease, they recognizes the specific sites in the DNA. Okay, this is the DNA long molecule. Okay, and they recognize which site they recognize and cut at that point and cut at that site. Okay, so Hind2 always cut at a specific, like other restriction endonuclease. This endonuclease also Hind2 also cut at a specific site after recognizing, and that part of region, a particular sequence of the DNA has 6 base pairs. Okay, 6 base pairs that is recognized, which is recognized by the Hind2, and it cut there. Okay, so that is the Hind2 makes blunt ends. Okay, so it cut there GTC GAC. Okay, there, and it cut the sticky ends. This specific base sequence is known as recognition sequence for Hind2. So for this particular restriction endonuclease, that is Hind2, this site where it is cutting and after recognizing, that is called the recognition sequence or recognition site for that particular restriction endonuclease, that is Hind2 for Hind2. Okay, so that is the. Now, besides Hind2, today we know more than 900 restriction enzyme or restriction endonucleases that have been isolated over 230 strains of bacteria, each of which recognizes a different recognition sequence. So, as I told you earlier, that each restriction endonuclease, okay, have got some its recognition site where it cuts. Okay, so like in Hind2, we found that it is a specific site where it is recognized and bind and then cut into. So for all the different, okay, so after the discovery of Hind2, there's so many. Nowadays, till date, we have got 900 restriction enzyme discovered, and we know that, and that has been isolated from over 230 strains of bacteria, and each which recognize a different, which has got each of which the 900 has got different specific recognition site where it cuts the DNA into fragments. So some of these are named as BAM H1 from the resource of microorganism, the Bacillus amylo coefficients. Okay, so that is the name of the bacteria from which this is the source. This is the source microorganism from which it has been the isolated. Okay, so the one which has been isolated from the Bacillus amylo coefficients is the BAM H1. Some of these about out of the 900. Some of these has been named, okay, here and given as an example, and the recognition sequence is also given. So GGATCC is the recognition site, GGATCC, and on the complementary site, CCTAGG is the region where it cuts. So it cuts between G and G and the G and G. That is the region. Now, in case of the guanine and guanine, and again on the opposite side also the guanine and guanine. Now, in case of the another one, the E-CoRI, the most common and highly and widely used one of the restriction enzyme, Escherichia coli RY3 is the source microorganism. This is the bacteria, Escherichia coli RY13 is the bacteria from which this has been isolated, and it has got its recognition site between the guanine and adenine, and that is the specific site and for binding. Okay, so this is the one. Now Hind3, Haemophilus influenzae, is the another one. Okay, this is the another source minus. It is a virus, it's not the bacteria. And here it is the we find this restriction enzyme is cutting at a specific site between adenine and adenine on the complementary side. Okay, so PST1 is another one that has been isolated from the Providencia stuartii, and this is has also got its specific recognition site as given in the table. SMAT is the another one, the Serratia marsensis. Okay, so this is also got the between the cytosine and guanine. Okay, and another one is the H pi. Okay, so this is a Haemophilus parainfluenzae. Okay, this is a microorganism virus. Okay, from which this has been isolated. Okay, and it has also got one particular and a specific site for recognition, and where it cuts the DNA into fragments. Okay, so this is in between the where it cuts the bonds of between the thymine and adenine. Okay, so there are some of the examples. Now the convention for naming these enzymes is that the first letter of the name of the comes from the genus, and the second two letters comes from the species or the prokaryotic cells from which they were isolated. So there are some kind of rules, and there is a specific pattern of naming these enzymes. Okay, it's not randomly named. Okay, but there is some kind of regulation, some kind of rules. Okay, naming these enzymes are there, and that is that the first letter. Okay. First letter of the names come from the genus. Okay, the second name or second two letters comes from the species of the prokaryotic cells from which it has an eco-RI. So we say like Escherichia coli. So the first letter comes the eco and co Escherichia coli. Right? So co, okay, coli, and Y I means one. So names based on the bacteria. Eco Escherichia genus, coli species. R means the strain, and the first identified order. Okay, one. R is called I here. Okay, one. So eco R1, say you can say, or I is the same thing. Okay, so this is how the restriction endonucleases are named. Okay, so there is a set of rules that is followed while naming these enzymes. Restriction enzymes belong to a larger class of enzymes called nucleases. Like I have been telling you since yesterday that the restriction enzymes are restriction endonucleases. So they belong to a class of enzymes called nucleases. Nucleases is a group of a class of enzymes. Okay, and this restriction enzyme belongs to the group of enzymes called nucleases. So these are the nucleases enzyme, and also called restriction endonucleases. So there are two kinds of restriction enzymes. Okay, one is the exonuclease and another is the endonuclease. When you say exonuclease, means it removes nucleotides from the ends of the DNA. Like while we are trying to chew sugar can, okay, and from the end, okay, and to drink the juice of that sugar cane, so from the one end. Okay, so exonuclease is cutting in the terminal region, okay, and endonuclease is cutting in the central, somewhere recognizing at the site and then cutting into the fragments. Okay, so that is the difference between exonuclease and endonuclease. Once again, I am repeating. Listen very carefully. That exonuclease will cut the DNA, the terminal, the last terminal region. Okay, and from where it starts cutting and into breaking. But in case of the endonuclease, it will somewhere in between, it will recognize one specific site where it cuts into fragments. So that is the difference between the exonuclease and endonuclease. Okay. Now here you can see the endonuclease activity. It's also called molecular seizures because these are the important enzymes that cut the substances into in the molecular level. So it's called the molecular incisors. Okay. So endonuclease activity, and you can see the exonuclease activity at the end, isn't it? The 3' end. Okay. So this is the how it exonuclease and endo. Each restriction endonuclease functions by inspecting the length of the DNA. First, it has to inspect and find. So that is the reason it has to inspect, because it has to find its specific site. So first it inspects, it finds, and then it recognizes its site of the for cutting. Then it cuts the DNA into fragments. So that is how the restriction endonuclease works. Okay. Now, once it finds its specific recognition sequence, it binds to the DNA and cuts each of two strands of the double helix at specific points in their sugar phosphate backbone. And you know very well the nucleotide, there is a sugar phosphate backbone is there. So different types of bones are involved in this DNA molecules, and one of that is the sugar phosphate backbone. Okay, so what happens when it inspects the DNA molecule in the sequences, it goes through and then it recognizes and binds, and then it cuts, okay, and a specific point, okay, that each strand and double at a specific point in the sugar phosphate backbone, and thus it is the DNA is broken. Each restriction endonuclease recognizes a specific palindromic nucleotide sequence in the DNA. What is palindromic? Palindromes are the group of letters that form the same words when they are read in both forward and backward directions. For example, if we say there are some kind of words, okay. In case of the base pairs pairing and the sequence, we get the GCA and ATA or ATGC like this, isn't it? But if we arrange, in permutation and combination, there may be some kind of word formed when we are reading from the first or the last. It will be read same word, okay. Like we have seen in the examples, like Malayalam. If you read from the left hand side, it's Malayalam, or if you read from the right hand side also, it will be Malayalam, like MALAYALAM, and from that side, the opposite side also, the right hand side, if you read these letters, it will be MALAYALM, like you have already studied from the left hand side. So that is we call it the palindromes or palindromic sequence. Here also, like we have seen, palindromic sequence in the DNA is the sequence of base pair that reads same on the two strands when orientation of reading is kept at the same, like this here. GATTC 5' to 3', and C again from 5' to 3'. So that means the one strand of DNA is connected to the two strands by hydrogen bonding. So one is antiparallel. It is antiparallel. DNA is antiparallel. That we study in the structure of DNA. So one is running from 3' to 5' and another is 3' to 5' in this way. So this sequence will be the same in case of the, so that will be GAATTC from 5' to 3'. And again on the complementary strand of the DNA also it will be GAATTC from 5' to 3' because they are running anti-parallel, okay. So anti-parallel is parallel position but running, okay, in a opposite direction. So the above sequence read the same on the both the strands, the two strands from 5' to 3' direction, and that is the we call that the palindromic sequence, palindromic in DNA, palindromic. This is also true if we read in the 3' to 5' direction, because if you read from the 3', it will be CTTAAG, or it will be again from the low, the complementary side, you can see CTTAAG. Okay, so that is from the what we call the 3' to so this is this kind of sequence where we have to read the same from both the sides. That is called the palindrome in DNA or palindromic sequence of the DNA. Okay. Now, commonly most restriction enzymes cut into two strands of DNA double helix at different locations. Okay, in different locations. What are commonly they both at different locations? So I will show you that also. Such a cleave is generally termed as segregated, staggered cut. Staggered cut means not straight, but I will show you that also. Restriction enzymes cut strands a little away from the center of the palindromic site between the same two bases on the opposite strands. Okay, because they are not exactly on the same line. So someone is like if you cut like this, cut and then like this. Okay, from here this way, it is slanting. So that is the staggered cut. Okay. So EcoRI recognizes 5' GATTC 3' sites in the DNA at cuts. It's between G and A. So GAA on the complementary side will be this side, not right in front of the GA, but it will be in front of the TC or TT like that. I'll show you that also. Here, see, you can see. So when it's cutting, you can see the G is cutting where? It is cutting there between the G and A. So G and A here. In case of the complementary strand, on the lower strand, you can see it has little towards the right hand side, and in case of the upper strand, it is little towards the left hand side. So here it is cutting into fragments, okay, at a little in a little different positions, okay. So this is called the staggered cut, okay, by the restriction. So these overhanging stretches are called sticky ends or cohesive ends, because they are ready to join with another fragment. Okay, so they are after cutting, these are called the sticky ends or cohesive ends. Okay, overhanging. Okay, now say these sticky ends. Okay, these are the sticky ends. Okay, now we'll be doing with another one. You can see that these are the sticky ends. Okay, the red one. Now, these are named so because they form hydrogen bonds with their complementary cut counterpart. This stickiness of the ends facilitates the action of the DNA ligase. So due to this stickiness, because why they have formed because they form hydrogen bonds with their complementary counterpart. Because when they are come closer to the another fragments, they will form hydrogen bonds with the help of ligase, and this stickiness helps the DNA ligase enzyme, that enzyme that helps the DNA fragments to join. So this stickiness helps the DNA ligase to join the fragments into the solid DNA. So hydrogen bonds formed like this, you can see here. Okay, so ligase and animation, and now this is covalent bond. You see here. Now you can see the DNA fragments. The ligase is there. The sticky ends are there. Now it's joining, and now it has ended. So hydrogen bonds has formed. So hydrogen bonds are there. So that is helping, and ligase is the enzyme that helps in the joining. Now this is how it is. Now can you calculate theoretically how frequent is the EcoRI or EcoR1, the recognition site sequence in a DNA? There are 4 types of nucleotides in a DNA. 4 types. Hence, the probability of okay. Now this means that you have got adenine, thymine, guanine, cytosine, etc. No, so you've got 4 types of nucleotides in DNA. So based on that, I told you at that time the permutation and combinations. Okay, so the probability that any one of these occurs at a place is 1/4, because we have got 4 types of nucleotides. Okay, a guanine, adenine, thymine, and cytosine. So assuming that occurrence of bases in adjacent position is random, the probability that a GAATC occurs is 1/4 to the power 6, or once in a stretch of 4096 nucleotides. So once, at least once after 4096 nucleotides, so it will get that sequence. So that is the by probability. Okay, calculating the probability by permutation and combination, it will form like this. Okay. Now, however, in reality, the location and frequency of such sequences in given genome is not random, but specific. Depending on the particular species, it will be not random, but it will be this pattern will be followed. In case of the specific wise, species wise. Okay. For example, in the human body or human Homo sapiens, or in other organisms, in particular species, depending on the particular, it has got a specific sites or a specific frequency or a specific location. Okay, depending on the species. It's not that in one of the human body it will have in different location, and other will have a different locations, and other will certain. It's not like that. If I have got at the 4096 position, after that sequence is coming, another human body will also have the same specific position after 4096. Okay, so it is depends on the species. Okay, it's not random. So restriction endonucleases are used in genetic engineering to form recombinant molecules of DNA, which are composed of DNA from different sources and genomes. So these restriction endonucleases we are using to cut the DNA and to join with the desired DNA, but desired gene, because that DNA will encode and express for a particular amino acid and protein that is of our interest. Okay, in genetic engineering. So this facilitates. This is opened a new era, you can say. Restriction endonuclease plays a very important role, and discovery of this restriction endonuclease has given a new dimension, I can say, you in the biotechnology subject. Okay, and or in the field of the biotechnology. Because without this restriction endonuclease, it is the genetic engineering is not possible. Now, when cut by the same restriction enzyme, the resultant DNA fragments with the same kind of sticky ends, and these can be joined end to end using DNA ligases. Now, when we are using the same kind of restriction enzyme or restriction endonuclease, it will have the same kind of fragments and it will have the same kind of sticky end. Why? Because it will recognizing a particular specific site. So wherever it will cut, it will have the same sticky ends. Okay, so that is the reason all the fragments will have the same kind of sticky ends, and these can be joined together end to end using the DNA ligases. Okay, so it is interesting to note that restriction endonuclease cuts phosphodiester bonds where DNA ligase forms the phosphodiester. Yeah, it's very interesting point. Yes, because we have got this tool. Okay, and this tool has facilitated so many different experiments, so many different discovery in the field of biotechnology. Okay, it is interesting to note that the restriction endonuclease are actually breaking down the phosphodiester bone. They are cutting at the phosphodiester bone, and then they are cutting the actually they are separating these the fragments of the DNA. They are attacking on the phosphodiester bone, whereas in case of the ligase, they are joining this phosphodiester bone, just opposite of the restriction endonuclease. Okay. So now we go through some important questions very quickly. That is, genetic engineering or recombinant DNA technology can be accomplished only if we have the key tools like I told you that the restriction enzymes, polymerase enzymes, ligases, vectors, and the host, and all of the above. Okay, because we need all this for recombinant DNA technology. Restriction enzyme for cutting, polymerase enzyme for forming the complementary strand. Complementary strand for the DNA, strand of the DNA, and ligases to join vectors to carry that vectors to insert the DNA of our interest, and then the host in which we are the inserting into the. Okay, so these all are required in DNA recombinant DNA technology. Okay, recombinant DNA technology. Now, in the nomenclature of restriction enzyme, the first letter of the name comes from the first the genus name of the organism, of the bacteria, okay, or species, strains, or family that comes from the genus. For example, EcoR1 or EcoRI, we know that the E has been taken from the Escherichia coli, where and CO has been taken from the species name coli. So it is a genus. Okay. Now, exonuclease cuts the DNA from the terminal region, and that is very easy, middle of the DNA or from the end of the DNA, or center of the DNA, or all of the above. It is from the ends of the DNA. Now, cloning vectors. What are cloning vectors? Vectors I have already explained yesterday. The DNA used as a carrier for transferring fragment of foreign DNA into suitable host is called vector. So, for example, we are taking one plasmid, one DNA, circular DNA from bacteria, and we are inserting our DNA of our interest into that particular plasmid DNA, circular DNA. So that becomes the vector, and then that vector we are inserting into host organism. So ampicillin resistance, okay, PVU1 and PST1, this reason, the green one, and the tetracycline resistance. These are the BAM, HI, and BAM. These we are inserting into the plasmid, and that is the origin of replication is there. So we are this is the vector where we have inserted the gene or the part of the DNA that is responsible for conferring and providing the resistance to the antibiotics such as ampicillin and tetracycline. Okay. Now vectors used for multiplying the foreign DNA sequences are called cloning vectors. Okay. So that means we are getting the number of copies. Okay. So used for multiplying the foreign DNA, the gene cloning with different vectors, bacteria, okay, and the virus. So both can take place, and we need to do this to get a multiple copies, okay, multiplying foreign DNA, and then we need to do some other experiment. Okay, so commonly used cloning vectors are plasmids or bacteriophages. It's not only the plasmid as we have seen in this diagram, even the viruses are also, especially the bacteriophages, cosmids, they are used for the multiplying and used as the vectors. Okay, so the site of the phage incorporated into plasmid, okay, and artificial chromosomes, etc. So here you can see the cytokine in opine right border, opine catabolism, origin application, and this has formed a new a vector, and this is the plasmid where we have included integrated some the DNA of our interest. Okay. Now in phage, lambda phage, head containing DNA, collar, tail, and the base plate. This is the structure of the bacteriophage. It looks like how it looks like. This has got a head containing DNA, and there's got some tail-like structure, and the collar is there, and then the base plate is there. The main portion is there, the head region where the DNA is contained. Others are the structure that provides the other functions. So EcoRI, and here cos site and SMA1 cos site. Okay, cosmid. In case of the cosmids. Okay, now this is how in case of the plasmid cosmid. This is the cosmids. Okay, bacteriophage cosmids. Okay, so this is how it's formed. Now, artificial chromosomes vectors are linear vectors constructed by adding centromere telomere, and origin of replication sites to DNA fragments. So artificial chromosomes are also prepared in the lab with the help of the constructed by adding centromere, telomere, and origin, because these are the part of the chromosomes that must be added so that to make it functional. Okay, so that is there. Now plasmids are extra chromosomal circular DNA molecules. So in bacteria, apart from the genomic DNA, apart from the main DNA, there are extra chromosomal DNA. Apart from the DNA that is present in the chromosome, there are some DNA, circular DNA that is present in the cytoplasm. So these are the plasmids. These plasmids are the extra chromosomal circular DNA and found in almost all bacterial species. So you can see the plasmids are there here shown in the blue color, and the bacterial DNA, main DNA that is present as the chromosomal DNA or genomic DNA. So apart from that, then if the cell replication takes place, you will find the two copies of these. Okay. So plasmids are inheritable and carry few genes which determine a variety of biological functions. So even the plasmids are having some genes, and they are carrying genes which determine a variety of biological functions. Okay. Now the advantage of a plasmid is that it is very easy to isolate and reintroduce into the bacterium, or that is the host. Okay. So because it is small, circular, and it is easy to isolate and easy to integrate the important our DNA or part of the DNA into these plasmids, and also it is easy to reintroduce into the bacterium cell, into the host cell. So that is the advantage it offers. Okay. Apart from the natural vectors, artificially restructured plasmids called the PB322 after Bolivar Rodriguez, the scientist, PUC 19101 after the University of California, are popularly used as the artificially restructured plasmids. Okay, so these are the done in the lab. Okay, artificial. Now plasmids and bacteriophages have the ability to replicate within the bacterial cells. Okay, and independent of the control of chromosomal DNA. This is the advantage it is offering, because when we have introduced that a particular segment of the DNA gene into the plasmids, okay, so that can multiply, that can replicate independent of the genes or DNA that is there in the genomic DNA. Okay, so that is one of the advantage that we get from this. Okay, even in the bacteriophage also, it is the independent of control of chromosomal DNA. Okay, so that is the reason. Okay. Now bacterial chromosomes, okay, transfer to E. coli, survive independent plasmid replication taking place. Okay, and then now it's formed now in the replica in the after multiplication we'll get the copies, because that has also replicated when the genomic DNA is replicating. Okay, but at the same time that also plasmid is replicating and forming of its copies. Okay, and that we get in this progeny. Bacteriophages, because of their high number per cell, have very high copy numbers of their genome within the bacterial cell. So here you can see the virus particle, okay, and virus DNA. Then we are actually using this bacteriophage technique for multiplying, getting the copies of the plasmids or the plasmids to get the proper copies, so we can do the biotechnological experiments, or we can utilize as per our aim and objective. Some plasmids may have only 1 or 2 copies per cell, okay, whereas others may have 15 to 100 copies per cell. Just imagine. So some plasmids may have 1 or 2 copies, okay, and some cells may have the 15 to 100 copies of per cells. If we are able to link an alien piece of DNA with bacteriophage or plasmid DNA, it can multiply its number equal to the copy number of the plasmid or the copy. So if we are alien or important one which have our interest within the DNA sequence of our inserting the alien DNA, so it will make the number of copies, like if the one plasmid is replicating into like 50 copies, so we'll get that the alien DNA after 50 copies also. Okay, so accordingly, so that will be the number of the DNA of our or alien DNA. If you are able to link alien DNA with bacteriophage or plasmid, it can multiply its number equal to the copy number of the plasmid or bacteriophage. Okay, So vectors used as present are engineered in such a way that they help easy linking of foreign DNA and selection of recombinants from non-recombinants. So we have to now that the vectors are used, okay, and at present, and they're engineered, they are prepared in such a way that the linking will be easy with foreign DNA, okay, because we need to link with the alien DNA. No? The following are the features that are required to facilitate cloning into a vector. And what are they? Origin of replication. This is very important. ORI, okay, or ORI. Because without origin of replication, if we are inserting, and then the replication will not take place, okay. Replication starts at the point, at the site where there is a called the origin of replication. So this is a sequence from where the replication starts, and any piece of DNA when linked to the sequence can be made to replicate within the host cell. So if this is there, so if it is inserted with this, then definitely the replication will take place, like this is shown. The origin, okay, daughter strand and parental strand, and the replication proceeds in the both directions, and this then there will be the direction and the termination of replication. Then there will be the two copies of the DNA or plasmid will be formed, produced, okay. So origin of replication is also responsible for controlling the copy number of the linked DNA. So this is, I told you yesterday also, because the origin of replication is important. Now if we want to recover many copies of the target DNA, it should be cloned in a vector whose ORI or origin of replication supports high copy number, okay. So that is another point. If we want to get many copies cloned, so we need to whose ORI supports high number copy, where there is high number of origin of replication is there, okay. So selectable marker. In addition to ORI, the vectors require a selectable marker which helps in identifying and eliminating the non-transformants and selectively permitting the growth of transformant, which will transformant and selectable marker. Okay, marker means the particular site, a particular region in the, okay, so selectable marker. So in ORI, the vector requires a selectable marker which helps in identifying and eliminating non-transformant and selectively permitting the growth of transformant, transformant. And the transformation is a procedure through which a piece of DNA is introduced in a host bacterium. So a transformation is a process, or you can say a method through which a piece of DNA is introduced in host bacterium. Okay, because we need to introduce or integrate the alien DNA into the host DNA. So that is another transformation. Now, normally the genes encoding resistance to antibiotics such as ampicillin, chloramphenicol, tetracycline, or kanamycin, etc., are considered useful selectable marker for E. coli. Okay, so these are the important markers, and thus is important. The normal E. coli cells do not carry resistance against any of these antibiotics. So normal E. coli cells do not carry, but when we introduce this, so this will be resistant to these antibiotics. But normally wild type of these Escherichia coli is not resistant to these antibiotics like chloramphenicol, ampicillin, tetracycline, or kanamycin. Okay. Now cloning sites. Okay. So in order to link the DNA, alien DNA, the vector needs to have very few, preferably single recombination sites for commonly used restriction enzymes. Okay, one single recombination site should be there. Okay, so that it will be used commonly used for the restriction enzymes. Now, the presence of more than one recombination site for the same enzyme within the vector will generate several fragments, which will complicate the gene cloning. Then it will be confusing there. So we have to choose. So in order to link DNA, the vector needs to have very few, preferably single recombination sites. So that will be easy. If there is so many fragments, then it will be easy, because we need to cut. If you want to join, there is a long a thread. Okay, it's a long thread. Okay, and you want to join a piece between in between. You need to cut in the center and then in. So if you cut it in three places, then it will be a problem. You want only two ends, only one break, or you need only one few break or one break. One cut, so to divide into two fragments, because we need to, for example, I'll draw a diagram here. You can see just here. So if this is the one, okay, and we need to cut here, okay, and we need to insert it here. So if it is a cut here, so many sides, then it will be a problem. Then we have to join it. We don't need those cuts. We need to only one cut. to introduce, to integrate, to join one alien DNA is one cut is sufficient. Okay, between one cut, when it is a long DNA, okay, and it's cut. But if it is circular, also it is cut one part. So that cut part has two ends now, and we join in that area, in that region. Then and then we, after joining, it will become again it will be complete circular DNA plasmid. So that is the reason we, in order to link alien DNA, reactor needs to have very few, preferably one deconjugation site. So that will facilitate our work and it will be easy. Otherwise, it will be problematic. Okay, now you can see here. Okay, the OD, multiple cloning site, okay, and DNA ligase. Okay, then the molecular weight. The cloning vector should have low molecular weight. Then it will be easy. Okay, vectors for cloning gene in plants and animals. The bacteria and virus transfer genes into plants and animals and thereby transform eukaryotic cells and force them to do what the bacteria and virus want. That is it. So what happens when the bacteria and virus genes can be, genes into plants and animals and thereby transform the eukaryotic cells and force them to do what bacteria and virus want. They want. Okay. So, for example, Agrobacterium tumefaciens, a pathogen of several dicot plants, is able to deliver a piece of DNA known as tDNA to transform normal plant cell into tumor and direct these tumor cells to produce chemicals required by pathogens. Okay, so this is how the biotechnology works. We are using this as we are inducing the tumor, and this tumor is directing the cells to produce chemicals, okay, required by pathogens. Agrobacterium tumefaciens, okay, we introduce a plant cell and then it is integrated and then the opines is produced and auxin, okay, and auxin cytokines. Okay, so this is how biotechnology works. We are inducing the production of these particular chemicals, okay, required by pathogens. So now, similarly, retroviruses in animals have the ability to transfer normal cells into cancerous cells. So, retroviruses, and this is the tumor-inducing TI plasmid aggregation has now been modified into a cloning vector such that it is no more pathogenic to plants, but still it is able to use mechanisms to deliver genes of our interest into a variety of plants. So nowadays, what happens? A tumor-inducing TI plasmid aggregation has been now modified into cloning vectors. Okay, but and it is now no more pathogenic now because we have biotechnology changed. Okay, and now similarly, retroviruses have also been disarmed and are now used to deliver desirable genes into animal cells. So retroviruses, they have been disarmed means we have biotechnology made them non-harmful, okay, and non-infective, and but we still use that to deliver some desirable genes into animals. Okay, but now competent host for transformation with recombinant DNA means the DNA is hydrophilic molecule. Okay, it is a water loving. Okay, it can be not repelling, it's not water repelling, so it can pass through the cell membrane. Okay, in order to force the bacteria to take up the plasmid, a bacterial cell must first be competent to take up the DNA. So bacterial cell must be competent to take because it is not taking it is not the DNA molecule is not hydrophilic, and the bacterial cell is not taking this DNA, then it will be a problem. Okay, and we will not be able to insert that. Okay, so that is the. Now this is done by treating them with a specific concentration of divalent cations such as calcium, which increases the efficiency with DNA, which enters the DNA bacterium through pores in its cell wall. So there are some pores in the cell wall that helps. Okay, so this is done by treating with a specific concentration of divalent cation such as calcium ion, okay, which increases efficiency with which DNA enters the in bacteria through pores. Okay, so when this is treated with calcium ion, okay, specific concentration calcium ion, so that helps, that facilitates the entry of the DNA to the pores that is present in the cell wall of the bacteria. Okay, so that is how it is making the entry into the bacterial cell by treating with the calcium ion. Okay, because that will help the pores are there at the cell wall, so the DNA can enter into the bacterial cell through that pores by treating with the calcium ion. Now, example of a restructured reactor is the plasmid, cosmid, bacteriophages. All of this is the cosmid. Okay, this restructured reactor. Now, in order to force the bacteria to take up the plasmid, the bacterial cells, plasmid, the bacterial cell must be potent cell, competent, totipotent, or impotent. If the competent, okay, that we have already studied. The most important feature of the reactors which eliminates transformants and from non-transformant is the origin of application on the cloning site or selectable marker or the molecule. It's a selectable marker, okay, selectable marker, which I already explained in the previous slides. So this was all about the recombinant DNA technology. Okay, how we use to form a recombinant DNA, how we use the different tools in biotechnology to form a recombinant DNA. Okay, so this was all about the DNA technology in the biotechnology, and now tomorrow again we will meet with another topic. So after this class, I'll send you the DPP that you will find in your academic booster. Download that and solve the answer and send back to us. Okay. So thank you for joining today and listening. Tomorrow again we will meet at the same time. Till then, goodbye and take care.